When we talk about systems biology, we're really talking about a holistic, data-driven view of physiology, of human health and disease. And when we study complex diseases like cancers that are heterogeneous, they're dynamic, and they arise from multiple perturbations throughout many different biological systems, throughout many different levels of organization within our body. Now, that's a pretty dense statement, so let me unpack that a little bit. So when I talk about heterogeneous, so cancers are heterogeneous in the sense that not only is each cancer unique, but even within a cancer, even within a given tumor, for example, there are different complexities that arise, and there's different subpopulations of cells that might be resistant to one therapy or another, and so that's part of why we need a systems approach. These diseases are also dynamic, so they change over time, and that's one of the things that we're interested in looking at. If we can really understand how we move and progress from onset through the progression of the disease through the treatment and diagnosis, then we can have a better interpretation and a better understanding of how to treat those diseases. And finally, I talked about different biological networks. And so certainly there are cancers that do target very specific areas and maybe arise from a single mutation. But generally speaking, there are it's not just one network. There's lots of different networks that get perturbed. And we're not just talking about on the molecular level either. When we are interested in looking at this holistic view, we're interested in looking at really the multi-scale nature of the human body. So we're interested in starting at the individual molecules and genes all the way up to organ systems and how these different networks within our body interact. And so ultimately, the systems approach is really about striving to encapsulate all of these networks together, to study longitudinally over time, to study the dynamics, and also to therefore uncover some of the complexities in the heterogeneity of different cancers. And so how do we do this, right? We advocate for a systems approach that uses lots of different technologies to profile many different modalities within physiology. So ultimately what we're trying to measure are phenotypes. So phenotype is biological feature. So hair color, eye color, these are phenotypes, as is the existence of a specific protein or maybe the amount of that protein. These are all different phenotypes. And so our goal is to phenotype an individual. And ultimately we want to measure lots of different phenotypes all at the same time, all in the same person. And if we measure enough of these, and the goal here is to really deeply phenotype an individual, and this is a phrase we'll kind of come back to, is this idea of deep phenotyping, then we can start to have a really holistic view of an individual if we measure a lot of different modalities and different features within a network. And so us biologists, we really like to use the suffix omics. So what is omics? So as economics is the study of money and economies, genomics is the study of the genome and all of the genes, proteomics is the study of proteins, and so we start to collect all of these different omics measurements to look at genes, proteins, metabolites, the small molecules involved in energy metabolism in the cell. And also not just molecular profiling, but also health profiling of an individual. So we like to collect health data, clinical labs, even kind of more recent push in the field is the exposome. So all of the different things that we're exposed to in our everyday lives, whether it's pollution or something to do with air quality, or whether it's all of the bugs and microbiota that we encounter just living our daily lives. And so that's this systems approach is really kind of taking this holistic view, and the way that we go about doing this is to try to use deep phenotyping and all of these different omics technologies to learn as much as we can and profile it individually kind of on a global level. As a researcher, I'm most interested in making these measurements and everything, but it's also very important to keep in mind the practical considerations of down the road making things that are clinically actionable and able to be implemented in the clinic. Generally speaking, we like to think that there's a lot of information and data that are available in 4 bodily fluids. That would be saliva, urine, spinal fluid, and the blood. Now, I think all of these could carry a lot of information, could be really informative. We really focus in on the blood. Lee Hood has said for years that the blood is a window into health and disease. And the idea is that blood is the only tissue that interacts with all of the other tissues and organs in the body. Therefore, any changes that might occur in any individual organ, say your liver, would be reflected in the blood. So if there's some perturbation, some change, some disease that affects your liver, the way that the liver interacts with the blood, different proteins that get secreted, these kinds of interactions, we're able to measure that in the blood. And of course, your blood is intrinsically accessible. Getting a blood sample from someone is fairly certainly routine part of clinical care, but it's also not a huge burden on the patient. And that's obviously a very practical consideration, is also how much matter do we need in order to make some of these measurements. Technologies are constantly improving, but we still need to have a certain amount of matter in order to make a measurement. And so that's a large part of where these technologies are going, is requiring smaller and smaller samples, because from just a single vial of blood, there's only so many measurements we can make. Because if there's just not that many of a particular kind of protein in that vial, we might need that entire vial of blood in order to make one measurement. But if we're interested in measuring a lot of things, then we might need to add more and more vials of blood. So that's mainly where our focus is to try to learn as much as we can about an individual by profiling the blood. As a data scientist, my answer is measure everything, right? Give me all of the data you possibly can. But that's not practical for a couple different reasons. The first is cost. It's expensive to make a lot of these measurements. And so while it's important for researchers, particularly because we don't know right now what is the best thing to measure, we're still in this stage of understanding and kind of uncovering a lot of this. So we need to measure everything that we can so that we can reduce the dimensionality of these massive data sets, of this big data that we have, and really zero in on what are the most important things. The other practical consideration is also, again, the burden on the patient. We don't want to have to take out a blood bag's worth of blood from an individual. We'd like to be able to just take a few small blood measurements. I think the ideal kinds of assays and technologies would be a finger prick, for example. But certainly taking a couple vials of blood from a patient, as would be part of routine clinical care every time you go in for a physical or pre-surgery or something like that. I think that's reasonable, and that's really where we're trying to get on the research side, is trying to reduce down the number of measurements we make. And as to your question about which measurement is most important, a lot of that really comes down, I think, to what specifically we're interested in looking at. That's kind of a vague, broad answer. I think it all starts with the genome. The genome contains all of the information that makes each of us unique. On top of that, then we could start to layer on some of these other technologies. The proteome, all of the proteins in the blood, is one of the most important measurements to make, because the current functionality and the current capabilities of your body is represented in the proteome. So the proteins are the compounds and molecules within your body that actually perform different functions. And so getting a snapshot of what the proteome looks like is really getting a snapshot of the functional state of your blood. A lot of active research has been using metabolomics to great effect. There's been some really outstanding research articles that have come out of ISB recently using the metabolome to uncover and understand what's happening in the gut microbiome. That's some research by Sean Gibbons and Nathan Price, and a couple of outstanding postdocs, Tom and Noah. So again, it kind of depends on what you're interested in studying, but I think it all really starts with the genome. I mean, we're certainly not the first or the only ones to think of personalized medicine and to think of personalized therapies. And the ultimate goal there is to get the right therapy and the right dosage at the right time. Deep phenotyping is a way for us to, again, take this holistic view and really kind of narrow in and target each of those different aspects. The dimensionality that we really advocate for is the use of deep phenotyping to define a baseline for each individual. So the goal here is to move away from population averages and toward what we in the science world call an N of 1 approach. So N is just the variable that we use to indicate sample size. So N of 1 means a sample size of 1. An N of 1 approach is essentially how each of us kind of thinks of ourselves, and that's the approach each of us takes to our own health in our everyday lives, right? I mean, I am an N of 1. I don't eat certain foods because I know that it upsets me or makes me sick. If I get motion sick, I'm going to take Dramamine before I go on a long flight. So each of us already kind of think of ourselves in this N of 1 approach. But our healthcare system uses a lot of population averages, mostly due to necessity. And so deep phenotyping is a way for us to move away from these population averages, where instead of me going in, getting a blood test, and getting compared against some population average, if I have deep phenotyping and we have all of these measurements for me over time, I'm able to compare against my own baseline. And that helps provide a much deeper understanding of when am I starting to deviate from a healthy state. And those deviations start to indicate the formation of disease. And so one of the kind of complications or arguments against an N of 1 approach is that it's really hard to build any kind of statistical confidence in an N of 1 approach. And that's absolutely right. But when you aggregate a bunch of N of 1 studies together, and they're all performed in the same way, and you have the same measurements, then we do have a large enough sample size to perform meaningful statistics and get the necessary statistical power in order to really learn important insights from all of these deep phenotyping N of 1 studies. Especially in the context of omics technologies and deep phenotyping, it's really the cost of these measurements that has gone down. Just this past summer, the cost of sequencing a human genome, the whole genome, dropped by 40%. It dropped from $1,000 to $600. We expect these prices to continue to drop to the point where they're getting closer to the point where they can be included as part of routine clinical care, as opposed to only for research studies or clinical trials, that sort of thing. One of the most exciting developments, particularly relevant to cancer, is the development of new single-cell technologies. Now, single-cell technologies themselves, and I'm talking about, instead of profiling populations of cells, I'm talking about measuring an individual single cell and being able to measure different compounds and molecules within that single cell. Those kinds of technologies are not new. Rather, it's the omics dimension. It's the ability to profile at the genome scale and to be able to measure thousands of DNA transcripts or dozens of proteins or on the order of a dozen metabolites. Those are the technologies that are really improving. And with respect to cancer, these kinds of technologies are particularly important because of the heterogeneity, and particularly the intratumoral heterogeneity. So again, this is this idea of within a single tumor, there's different subpopulations of cells, and these different cells have different properties. They might have different metabolic functionality, they might have different resistance to different types of drugs. And so if we're able to use these developing single-cell technologies to better understand how each of these single cells are functional, what their different phenotypic properties are, that just allows us to develop even better therapies and to really start to understand then how can we personalize the therapies down to each person's cancer. If we can take a sample from a tumor, learn as much as we can. If one drug only kills 70% of the cells in a tumor because there's multiple subpopulations, single cell technologies are going to help us get past that to 80, 90, 100% of effectiveness of different therapies. Certainly immunotherapy and everything in that area is one of the most exciting paths forward for the development of cancer research. And there's a couple kinds of immunotherapies. One is aimed at boosting our own immune system to help better fight cancer. And these kinds of therapies are being pioneered by lots of people in the field. There's some fantastic work being done here at ISB with Jim Heath, our president, in his lab, and of course others. And the idea here is that deep phenotyping, this idea of, again, measuring many different things longitudinally over time, can really help improve how we're able to personalize these immunotherapies. So if we have the one type of immunotherapy that helps boost the immune system, the other major type of immunotherapy is one in which we're able to engineer cells that can help fight an individual's cancer specifically for that individual. And how do we do that? We have to know a lot about an individual in order to be able to personalize that therapy. We have to know how the immune cells interact with foreign entities, with the tumors, and all of these kinds of things. And our systems approach, again with deep phenotyping, this is our strategy to really building personalized therapies. And ultimately, it will also help develop multi-drug and combination therapies, which likely will be a really important part and play a vital role as we attempt to take cancers from fatal diseases to chronic disease. The key point there is really it comes down to these technologies need to be affordable and they need to be accessible. And most of that comes down to the cost of these different measurements, all of these different deep phenotyping technologies. Really one of the exciting advancements here is the development of point-of-care diagnostics. So these range from sequencing devices that are the size of a cell phone and can measure a genome just from that device, and you can collect the sample from a patient right there, to imaging technologies, these point-of-care ultrasound devices that plug into an iPhone, and using an iPhone, a physician or a nurse or a tech can take an ultrasound image of an individual. These sorts of technologies provide the kind of deep phenotyping data and all of the information that really is needed to be able to start to translate this into the clinic. And so really it comes down, again, to the affordability of these, and as the affordability generally increases and the cost goes down, these kinds of technologies are going to be more accessible. There's a lot of different ways to go here. I think I'll focus on three. We'll keep it at that. There's more that we talk about in the paper, but the first one that I would suggest is one of the goals for the next 10 years really has to be defining a metric for what makes a well person, a healthy person, well. Right? So if we brought in 1,000 different people right now and we did this deep phenotyping and we took all of these measurements for these otherwise healthy people, we're going to get 1,000 different answers for what is healthy because we're all unique. A phrase I've heard is that healthcare is one of the only industries that doesn't have its own gold standard. When we go into the doctor, we're compared against population averages because I don't have a gold standard for what does it mean for me to be healthy or for you to be healthy. So that's one of the absolute key points that we really need to focus on is how do we quantify wellness? One of the ways to do this perhaps comes down to this concept of defining your biological age. So your chronological age is, how many birthdays have you celebrated? Your biological age is a developing research field that attempts to understand all of these different modalities in your body. How do they contribute to how your body functions? There's been some fantastic work, again, out of ISB here with John Earls and Nathan Price and Lee Hood, asking these exact questions and trying to understand how does your biological age, how can that be used to quantify how well you are? And again, it's important because if we're able to quantify our wellness, then we're also able to quantify when we're not well. And this goes back to this N of 1 idea, where if I have my baseline and I understand what my wellness is, I can start to detect deviations and as we move away from there. The second point that I would discuss is it's going to be important moving forward, and particularly to your last question about the affordability of these kinds of technologies and this kind of approach, is to identify those individuals who are at high risk for developing different cancers. So if we're able, potentially from the genome, or maybe from the genome and some other measurements, to identify whether an individual is at higher risk for developing a brain tumor, for example, then we can have that individual go in and start getting an annual MRI, for example, whereas giving every single person an MRI annually is certainly not feasible on many levels. But if we're able to do that for a targeted group of individuals, then we could really improve the chances of diagnosing these kinds of diseases. And ultimately, that's really what it comes down to, is diagnosing these kinds of diseases, these state changes, as early as possible. Because the unfortunate truth is just that because these kinds of diseases like cancers are so complex, as the disease progresses and it affects all of these different networks throughout the body, by the time, if we were able to cure even that original disease, there's so many other networks that are perturbed that curing the original disease doesn't really solve the problem, because now there's a whole string of other issues. So it really comes down to being able to identify individuals at high risk so that we can diagnose earlier when we start to deviate from a healthy state. And finally, I think just continuing the ongoing quest to better understand and elucidate the biological mechanisms that underlie the onset and the development and the progression of cancers. I mean, certainly that's what we've been working on for a long time, and I think that this systems approach that we're advocating for is certainly one way that we can move forward in that area to better understand these mechanisms, because if we understand the mechanisms and we understand how it works, kind of going back to that Richard Feynman quote, then we can start to engineer solutions and viable therapies and treatment options. Great. I've been at ISB for a little over a year now. My position is really aimed at translating the basic science research that happens to the clinic and making clinically actionable knowledge. So translation is kind of a buzzword that we use in the life sciences. It's just applied research. And so most of my doctoral work certainly had a little bit of a translational spin, but a lot of it was very basic science, looking at underlying mechanisms and studying biochemistry. And my role here at ISB is allowing me to translate this to the clinic and to study clinically actionable diseases and health states and these kinds of things. And so this fellowship is an opportunity for me to work closely with the physicians at Providence St. Joseph Health through collaborations with ISB, and to start to build these collaborations and work directly with physicians, which is so important for us researchers, is to be grounded and make sure that the research I'm doing, as interesting as it might be, that it remains applicable. And it's easy as a researcher to kind of go down a rabbit hole and just spend a long time studying something that's really interesting, but it may not have immediate clinical impact. And so that's really kind of the goal of the program. Moving forward, I'm interested in studying cancers, and really my research focus is just this systems approach, not necessarily specific to cancer. One of the other areas that I'm interested in looking at is the diagnosis of prediabetes and type 2 diabetes, and really, again, it all comes down to personalized medicine and using deep phenotyping to move forward and to personalize blood tests specifically. So it's estimated that about 2 billion blood tests are performed annually in the U.S., and there's been some reports that these blood tests inform the majority of clinical decisions. But these blood tests are all compared against population averages, which again really goes against how we think of ourselves and this personalized medicine. And so in the context of diabetes, we're interested in using the genome and some other omics measurements to interpret and personalize these blood measures, so that when you go into the doctor and as part of your physical you get this blood test, you get back a piece of paper with 2 columns of numbers on it. This left column is my numbers and the right column is the healthy range. We want to be able to have that healthy range not be some population average. We want that to be your healthy range, so that when you go into the blood and get a blood test next year and your number is outside of that range, then we can help use that to target.